Let's take a look at active transport. With active transport, we have primary active transport and secondary active transport. Your primary active transport is going to move against the gradient. So we're moving against that concentration gradient. Instead of going from a high concentration to a low concentration, now we're going from a low concentration outside the cell to a high concentration inside the cell. In order to do that, we need some energy to drive it in. So it requires ATP and it requires a carrier protein. With the carrier, it depends on what kind of class it is. We have a P class, which requires phosphates, and that's why it's called P class. Like our calcium pump, our sodium potassium ATPase pumps are P class pumps, but they transport a bunch of different electrolytes. Sodium, potassium, calcium, hydrogen pumps as well. With our sodium potassium pump, which we said are your ATPases, it's one example of those. With our sodium potassium ATPase pump, we have 3 sodiums that are being driven out of the cell, and it's going to be replaced with 2 potassiums that are coming into the cell. So this happens in nerves. This happens just to maintain cell volume. Anytime there's a slight change in cell volume, sodium potassium ATPase pump kicks in because sodium will push water along with it because sodium binds water onto it. So it's going to maintain cell volume. Other pumps that we have F-class pumps that are located in mitochondria, and those are known as your proton pumps. They're important for pumping hydrogen ions. Other ones that are important for pumping hydrogen ions, that line drawn to that means, another proton pump, are your V-class, which is located in your lysosomes, and that's why your lysosomes contain acid hydrolases for digestion. To maintain the acidity, we have hydrogen ions that are going to be pumped in using those V-class pumps. And then your ABC class pumps pump drugs or transport drugs. There's nothing else to know about the ABC class pumps. I don't care that you know much about them. Our secondary active transport is driven by the ion gradient that comes from our primary active transport. In most cases, oftentimes it's going to be sodium that's going to help drive this part of it. With secondary active transport, we can either have symport or antiport. So when we have symport, that's movement in the same direction, whether it's sodium and glucose going inside the cell, same kind of thing, or we have movement in the opposite direction. Maybe you have calcium going in and another compound going out, or vice versa. So symport, same direction, antiport, opposite direction.